This is a lecture on resistance to and tolerance of pathogens and wounding agents by forest trees. I struggled a little bit with what to call this lecture. I started out with disease resistance. Well, resistance really isn't enough because sometimes resistance sort of implies that you repel or stop, for instance, infection with the pathogen. And sometimes the infection happens and you just have to tolerate it. So we need both resistance and tolerance in there. And then disease, which is caused by pathogens, is not the only factor that impacts trees. And a lot of these mechanisms actually help trees to tolerate other damaging or wounding agents, including insects, including mechanical damage, breakage from storms or whatever, or fire as an example. So this was the best I could do: resistance to and tolerance of pathogens and wounding agents. So have a couple of examples here. Obviously fire here. This is a tree that has been infected with a decaying agent, probably a fungus. Certainly insects can be a factor. Other herbivores also, even large animals like birds, porcupines, herbivores like elk or deer. All of those things are wounding agents. And then this is an example of a tree that's been infected with the fusiform rust fungus. So diverse things that can impact trees, and these adaptations are important and also somewhat unique to plants, because plants and trees are sessile. They can't move. So trees can't run away from a fire or from an herbivore, so they have to have mechanisms to resist or tolerate those things. Another important element of these is that these adaptations that we're going to talk about in this lecture are probably some of the most important elements that contribute to trees' longevity. So especially trees' ability to compartmentalize damage or to compartmentalize attacks from pathogens are important in enabling them to continue growing and continue living for really long periods of time. So all of these things are sort of unique to plants and to trees, and one of the things that makes trees special. One of the first questions we might ask ourselves is, what is the ultimate function of tree defense or tolerance to pathogens and wounding agents? What has sort of driven natural selection for these traits? And really, in the end, some of the primary things that are being protected are the active sapwood of the xylem that transmits water and contains nutrients, the vascular cambium, which if it goes away, the tree will die because it can't produce the transport tissue of the tree, the xylem and the phloem, and then the phloem, which contains nutrients and sugars and therefore is a target for herbivores and pathogens. So protecting those elements is really important. You could probably, add protecting leaves as a secondary function, in that leaves also are, as the source of food for the tree, protecting them is probably also a secondary driving factor for the evolution of these traits. Most of what I'll be talking about in this lecture refers to stem and wood-focused defense mechanisms, but we'll hit a little bit on leaves as well. We can broadly class or classify defense or tolerance mechanisms into two broad categories, constitutive and inducible. So constitutive defenses are adaptations or traits that are already there. They're essentially always there so that when damage happens or when a pathogen attacks, those defenses are there. Whereas inducible traits are traits that are a response to an attack or to damage and happen after the insult or the damage. So constitutive and inducible. And then both of those, both constitutive and inducible responses can include both structural or morphological elements. So these are things having to do with cells, how they're arranged, and so on, as well as chemistry, chemical elements of defense. So those are very broadly how you can chunk out or categorize these traits, and that's how they're arranged in the lecture outline. When thinking about constitutive or preformed anatomical defenses, probably the overarching or ultimate one is that trees are highly compartmentalized to start with. If you look at, for instance, a cross-section of a piece of wood, you can almost see the compartments in that cross-section. So this is a diagram from Alex Shigo, but basically this is a cross-section of an angiosperm showing growth rings. So this is one growth ring here, and he's pointing out that those growth rings themselves form a barrier or a wall of a compartment. And then in addition, the ray cells, which run radially, form another wall, so that the cells within those boundaries of growth rings and ray cells are basically somewhat separated from adjacent compartments. So even just through the normal growth of secondary xylem in trees, those structures already provide some degree of compartmentalization and therefore some protection from spread of, for instance, pathogens from place to place in the tree. So that's almost the ultimate initial constitutive structural element, is that existing compartmentalization. Shigo included this, neat diagram or model made out of plastic where he shows the vessel elements or tracheids, and they're essentially bundled within those compartments that we referred to, bounded by growth rings and by the rays. So that's a more stylized or explicit demonstration of that compartmentalization. In terms of constitutive morphological defenses, the outer bark or periderm is really the first line of defense. When we look at a tree, the first thing we see is the bark. That's the outer covering of at least the woody part of the tree. So that provides a physical barrier to penetration by insects, fungi, herbivores, or whatever. The outer bark is formed by the cork cambium, and it usually has multiple layers, and those outer bark cells are mostly dead, They're highly lignified. So that lignin is not easily digestible. It makes those cells tough. Those cells are often encrusted with calcium oxalate crystals. And we'll talk about those a little bit later. You'll see some electron micrographs of calcium oxalate crystals, but essentially those are, almost sharp mineral crystals in the cell walls that basically make those cells tough and unpalatable. And then these often contain high concentrations of phenolics. So, we're slopping over here into chemical. And that's just a good reminder that when we categorize things in reality, generally categories work together. So clearly bark is a physical barrier, but that bark also has chemical traits that also make it a good defensive tissue. Another preformed defense mechanism that's important is the cortex. Now the cortex, if you think about primary growth from an apical meristem in a branch or a stem before secondary growth has occurred, most of that tissue is cortex around the vascular bundle, even in conifers or dicots. But as secondary growth occurs, that cortex sort of gets pushed out. So we can see here that the cortex is this space, this tissue between the secondary phloem and the periderm or the outer bark. So this cortex zone is important, especially in young trees, because it's another physical barrier, but also because it contains a lot of resin canals, especially in pines and some gymnosperms. And those resin canals are quite important for repelling pests. So that's the cortex. This is a cross-section on an older pine tree. This is a 4-year-old pine tree, and it still has some cortex here, and you can see those resin canals in the cortex. As the tree gets older, that cortex becomes less apparent, and the secondary phloem becomes a more important tissue for defense. So that brings us to the secondary phloem. So just to orient you here, this is a cross-section of a stem, and they're contrasting pines and non-pines here, but really there's a lot of similarities. So we have xylem tissue here, secondary xylem, and then the cambium, and obviously the cambium produces xylem to the inside and phloem to the outside. So this is a really zoomed in view of phloem tissue, and there are some important elements here. I did want to point out they're showing the periderm here. This is the outer bark or periderm. They don't designate or show the cork cambium, but the cork cambium is another meristem that's producing the periderm out here. But we're focused on the secondary phloem here. And there's a few elements that are really important in the secondary phloem for defense, especially in pines and other conifers. And one of those is the presence of resin canals. So there are resin ducts or resin canals that go both in the xylem and into the secondary phloem. And those resin canals are lined by epithelial cells. So those are living cells that line these ducts. and those epithelial cells produce resin. So resin exists in these resin ducts or resin canals under a little bit of pressure, so that when an insect, for instance, encounters one of these resin canals, they may be pitched out or pushed out. And I'll show you a picture of a pitch exudation on a stem later on. So those resin ducts or resin canals are pretty important defensive structure. Another important structure are lignified sclerenchyma cells. They're also sometimes called stone cells. So these cells exist in the secondary phloem, and these are basically heavily lignified cells in the secondary phloem. They're heavily lignified, so they're tough, so they provide some toughness to the secondary phloem, and they're also not easily digestible. So those lignified sclerenchyma cells or stone cells are an important element that sort of toughens up that secondary phloem. And then there's some other types of cells in the secondary phloem called polyphenolic parenchyma. So these are really abundant cells in the secondary phloem that contain phenolic compounds. So as their name implies, those polyphenolic parenchyma contain and manufacture phenolic compounds. Okay, so this is, another interface between anatomical and chemical. Okay, so those polyphenolic cells are a morphological trait, but they contain phenolics, which are antimicrobial compounds that they're also not palatable to herbivores. So those polyphenolic parenchyma are important elements in phloem that make phloem less palatable to herbivores. Now, obviously, also in the phloem are sieve tube cells, which transport sugars. So those sieve tube cells really aren't labeled on these diagrams, but all of these cells, are the sugar transport cells in the phloem, but they're not explicitly defensive. Those sieve tube cells are being protected by those polyphenolic parenchyma and those stone cells. Also in the secondary phloem are cells that have calcium oxalate crystals embedded in their cell walls. So this is yet another defensive mechanism where those extracellular crystals, this is an example of a calcium oxalate crystal, and you can see them here in these micrographs. Those are, again, just hard, sharp structures that sort of provide an additional layer of defense against herbivores or pathogens in these cells in the secondary phloem. Now, moving away from the secondary phloem and the cortex, there are many other anatomical traits that can be important for defense or tolerance. And one of those are leaf traits. So leaf traits like heavily lignified leaves or thick leaves or leaves with a lot of suberin. All of those things make leaves more difficult to penetrate or chew. Now, there's a lot of variation. Some leaves are quite thin and sort of tender, and therefore might be more vulnerable to herbivory, whereas a leaf like southern magnolia is heavily lignified, probably has a lot of suberin and therefore is more resistant to attack by herbivores. There are some cells in some leaves that contain thick-walled, lignified epidermal cells here, but also these hypodermal sclerenchyma, which again have thick cell walls and are basically another barrier to penetration either by herbivores or potentially even pathogens. Here's another defense mechanism. That doesn't look very inviting, does it? These are thorns on a honey locust. And so spines, prickles, and thorns are all sharp structures that repel or make plant parts less palatable or accessible to herbivores. Clearly a thorn, spine, or prickle is not much of a barrier to a pathogen, but certainly can be a barrier to an herbivore. And those three different types or categories of these sharp structures basically distinguishes where they come from. So thorns, which are shown in this picture, are modified or outgrowths of stems. Whereas spines are modified leaves or stipules. And then prickles is an outgrowth of the bark or epidermis. Now, certainly that honey locust picture looks like an outgrowth of the bark or epidermis, but I looked in several references and they referred to those honey locust outgrowths as thorns. So presumably that's some stem-related outgrowth. At any rate, all this sharp stuff keeps herbivores away. A final anatomical trait that I want to mention, it's really not anatomical, I'm just sort of throwing it in there as a category, but it's the microbiome. The microbiome basically refers to other organisms that are living on or in the tree. So you've probably read about the human microbiome and the fact that, for instance, some significant fraction of our biomass are other organisms. Well, the same is true of trees. We understand it even less. But there's a growing body of literature. This is sort of an emerging area of research that suggests that the microbiome may be important for imparting resistance to disease in trees and other plants. Now, probably the most well-studied microbiota are mycorrhizae, which are a mutualistic association between fungi and plant roots. And there's some thinking that that may help to impart resistance to disease at some level. Mycorrhizae have many other benefits as well, but that's one example. But there certainly is research that's showing that there are many, many categories of microorganisms that live on or in trees. So this is an example of just fungi, different fungal orders that live on roots, stem, and leaves of Norway spruce, as well as free living in the soil. But again, this is an emerging area of research, so stay tuned. This will likely become a really important area of research. The second category of preformed or constitutive defenses is chemical. So supposedly everything I've been talking to up to this point is structural or morphological or anatomical. But chemical defenses really, as I've already mentioned, interface with those anatomical defenses, but we can categorize them a little bit separately. So chemical defenses are just substances with toxic or inhibitory effects, usually on pathogens, but sometimes on herbivores as well. There may be some of these that are important for repelling herbivores as well. So one of those categories of chemical defenses are terpenes. And terpenes are the major constituents of resin. So there are monoterpenes, sesquiterpenes, and diterpenes, but together they form components of resin. So this is an example of resin that's being exuded out of a pine stem after a beetle attack. In this case, those terpenes in the resin canals under pressure, when the beetle encountered that resin canal, because that resin was under pressure, it actually pushed the beetle out. So that popcorn looking material there is actually globs of hardened resin after it's been pushed out of the stem. Another category of chemical defenses are phenolics. Now this is a very diverse category of compounds. There are thousands of phenolic compounds synthesized by different pathways, but things like flavonoids, tannins, other polyphenolics are all included in this group. And again, these have toxic or inhibitory effects. And then there are antimicrobial proteins that defend against microbes. So for instance, chitinase is an example. That digests fungal cell walls. So as you can imagine, that might be a useful chemical in defense against attack by fungi. Another example are beta-1,4 glucanases, another protein that digests fungal cell walls. There's another class of proteins called defensins that also defends against fungi. So there's a number, thousands of chemicals that are produced by plants and trees that help to defend against microbes and in some cases herbivores.